Okay, so now I have added in those bases. Remember, we started with this one, this thymine here. We started with this adenine, and we have to add away from that replication fork. So I've added the bases in all the way down, but I could not hook these two pieces together. So if you look here, I've got a piece of the strand, a fragment, if you will, that can't be added. DNA polymerase can only add if there's a base that's open over on this side and there's a 3' end. And when we got to this one, it had that open base, but when we got to this one, there wasn't one. There's nothing here for it to add to, so it has to stop. So in this case, I've got a fragment, and then I've got another fragment. These fragments, these pieces where the DNA polymerase on this side had to stop, move up the chain, restart, and go away from the replication fork, these fragments that are formed are called Okazaki fragments. And so DNA polymerase, again, its only job is to add new nucleotides. That's its only job. You know, it can only add it to the 3' end. It doesn't join fragments together. There's a special enzyme that will have to come in and join those fragments together, and that enzyme is called DNA ligase. And DNA ligase works down this side, and it looks for these kind of breaks, these chunks of these fragments. Now, usually Okazaki fragments, they're usually hundreds of bases long, but you know, because of this model, in the interest of time, we don't have them. So I'm going to color this one in blue, just so that you know that this was a fragment, that this one was a fragment, and this one was a fragment, and that we had to have a special enzyme come in and join these fragments. So let's write that special enzyme down on our enzyme list here real quick, so that we have that to study by. And this enzyme is called DNA ligase. And its job is to join Okazaki fragments. That's its job, is to join Okazaki fragments. So these are the basic enzymes that you have to, as pre-AP students, you have to know to be able to understand DNA replication. There's a couple other pieces of information that you have to know to be able to understand what's going on and answer questions correctly. And that is the fact that this side that has the Okazaki fragments on it, because it has to wait until enough of the strand is opened and work backwards, not backwards, but away from that replication fork, away from the part that has been split open by helicase. This side is called the lagging strand. So this side is lagging behind the other strand, the other side. And the other side then, because it's the one that gets done first, it's the one that can keep adding bases. It doesn't stop. The DNA polymerase does not start and restart. It just keeps on going. This side is called the leading strand. And when scientists have looked at, biologists and biochemists have looked at this process, you can actually see there is a difference. You can see the nucleotides being added on the one strand, and you can see the other strand is a little behind on adding its nucleotides. So this is called the leading strand. Now this is going to continue to work. This is going to continue to add bases until it gets all the way through. So again, once we get to a point where we can't add any more bases, helicase will come in and it'll finish cutting that hydrogen bond. DNA polymerase on the leading side will just keep on making the bases, keep on adding those things in. As long as it has a base to add to, a complement to add to, and it's got a 3' end, it's going to keep going. The lagging strand, it's going to have to wait until enough of it is split open to where it can back up the strand and then add back to this way. DNA ligase will come in just like it did here and add those fragments together. Now I want you to notice something here, and I'm not showing you the whole piece, I'm just showing you this one little section. The white, that was the original strand. So let me fold this over so you can kind of see that better. The white was the original strand. So this was our original strand right here. But after we've done replication, Watson and Crick proposed it. There were a couple other scientists who suggested that, who did the actual experiment to show that it did work. But these two sections are the old pieces. These blue here on our two new strands, so there's two new strands here. These blue pieces are the new nucleotides that were added in. This model, this concept of how this works is called the semi-conservative model. In other words, one strand is conserved, is kept, and a new strand, a new set of DNA nucleotides are put in, and so we semi-conserve the sequence of bases here, simply because we hold on to that one original template, and then we have our new piece that comes in and gets added. If this was going to be cell division, then one of these sets of chromosomes would go to one cell when the cell divides into two, and one would go to the other side. Again, please note, we start off with the two sides together. The first thing that has to happen is this has to be unwound, and then helicase comes in and cuts that that hydrogen bond. On one side, DNA replication, as soon as the primer is added, can get added immediately by DNA polymerase. The other side has to wait until enough has been moved open, or enough has been unzipped, so it can operate or add bases back away from that origins of replication, creating Okazaki fragments that DNA ligase has to come in and hook together. That's all for this lecture. Thank you.